Hello everyone, welcome to Biology Bingo. Today let's talk about pollination. Have you ever wondered why bees are attracted to flowers? Flowers look beautiful and are fragrant, but there's a reason behind this. Flowers actually help plants to reproduce. Flowering plants have seeds, carry the genetic information of parents and develop into a new plant. Flower. Flowers can be made up of different parts, but there are some parts that are basic equipment. The main flower parts are the male called the stamen and the female called the pistil. The stamen has two parts, anthers and filaments. The anthers carry the pollen. These are generally a lone color. Anthers are held up by a thread-like part called a filament. On the other hand, the pistil has three parts, stigma, style and ovary. The stigma is a sticky surface at the top of the pistil. It traps and holds pollen. The style is a tube-like structure that holds up the stigma. The style leads down to the ovary that consists of ovules. Other parts of a flower are petals and sepals. Petals attract pollinators and are usually the reason why we buy and enjoy flowers. The sepals are the green petal-like parts at the base of the flower. There are two types of flowers, unisexual and bisexual flowers. Sexual or perfect flowers have both male and female reproductive structures. The male reproductive structure is called the androecium and the female reproductive structure is called the gynoecium. Unisexual flowers, reproductive structure that is either functionally male or functionally female. In angiosperm, this condition is called imperfect or incomplete. Now let's talk about pollination. The transfer of pollen grains from the opened anther of a stamen to the receptive stigma of the pistil is called pollination. Each pollen grain grows and provides two male gametes for fertilization of an ovule. Types of pollination. Autogamy. It is a type of pollination in which the pollen grains from the anther of a flower is transferred to the stigma of the same flower. For example, can be wheat, rice and pea. Type is zytonogamy. It is a kind of pollination where the pollen grains from the anther of a flower are transferred to the stigma of another flower born on the same plant but at a different branch. It is seen in cucurbita. Xenogamy. It involves the transfer of pollen grains from the flower of one plant to the stigma of the other plant. This is the only type of pollination which brings genetically different types of pollen grains to the stigma during pollination. For example, papaya and maize. Now let's look at the agents of pollination. The agents responsible for pollination in angiosperms have been grouped into two main types. One is abiotic agents, other one is biotic agents. In abiotic agents we have wind pollination and water pollination, while in biotic agents we have mammals, reptiles, birds and insects. Outbreeding devices. Flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. Because the majority of flowering plants produce hermaphrodite flowers and are likely to come in contact with the stigma of the same flower, the continued self-pollination leads to chances of inbreeding depression. Some devices to prevent inbreeding depression are receptivity of pollen release and stigma is not synchronized. Either the pollen is released before the stigma becomes receptive or stigma becomes receptive before the pollen release. In some species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower, which will prevent autogamy. Self-incompatibility is a device to prevent inbreeding, which is genetic phenomena of inhibiting pollen tube growth on the stigma of the same flower. Another device to avoid self-pollination is to produce unisexual flowers. Now let's talk about artificial hybridization. Artificial hybridization is a process in which only desired pollen grains are used for pollination and fertilization. Here we have two things, emasculation and bagging. We know hybridization is a method of selecting breeding. Thus, anthers have to be removed from a bisexual flower before they release pollen grains. This step of removal of anthers using forceps is termed as emasculation. In the case of unisexual flowers, this step is not needed. Bagging is the protection of emasculated flowers from contamination by undesired pollen grains. Here the flower is masked by a bag, still the flower attaining receptivity. In unisexual flowers, bagging is done before the flowers are open. Emasculation and bagging ensures that the female flower is completely protected from contamination. Once the flower attains stigma receptivity, the desired pollen grains are dusted on the stigma. This is resealed for further developments. Hence, artificial hybridization ensures that the right type of pollen grain has been transferred to the stigma of the flower. In addition, the chance of fertilization is high. Through this approach, a variety of strains of crops can be developed and it improves the quality of crop with desired characters. Thank you. I have a few questions at the end. Please attempt it and write it in the comment box.